Hello students, today I would like to let you know about the role of mutation breeding for crop improvement. In this class, you are going to know about what is plant breeding, what are the principles of the plant breeding, what are the different methods of the plant breeding and different modern methods of the plant breeding. So, in this topic, actually I wanted to make you know about the plant breeding. What is plant breeding? Plant breeding is the applied science of the botany. You know that plants plays a vital role in providing food for all organisms, but when there is increase in the human population, there become scarcity for the food. So, several famines are there in the history of the human civilization. Then the plant breeding came into existence with the applications of the botany principles. So, our farmers are well known about the breeding principles. These conventional plant breeding practices are age-old practices, and they collected different types of the germplasms and they introduced the new varieties and they domesticated several varieties of the crops, and even they hybridized several crops also. So, these are all good age-old practices of the farmers, but recently our plant breeders, that is the scientists of the botany, they invented new methodologies in plant breeding by applying modern science tools like mutations, tissue culture practices, polyploidy breeding, etc. Usually in the plant breeding, there will be the creation of the new varieties or the exploiting the genetic variation for the introduction of the new varieties. So, there is a selection of the plant material, that is the selection of the parents would be there. So, after selection of the parents, there is a evaluation for their yield and growth. If we are satisfied with this growth and yield, then it will be released as a variety. This is the common practice in every method of the plant breeding protocols. Even in the introduction or domestication, or you take hybridization or mutation breeding or polyploidy breeding, you take whatever the protocol, this is the common practice. So, there is a selection of parents and evaluation of the parents for their growth and yield. Finally, there will be a release for the crop variety. So, finally, the plant breeders distribute the new variety to the farmers and released as a new crop. So, these are the modern methods of the plant breeding. Here you are watching the modern methods developed by the plant breeders nowadays. One is a mutation breeding, polyploidy breeding, and other biotechnological methods are also available. But today in this class you are going to know only about the mutation breeding. So, you all know that there are several varieties you are observing even in the market. You are observing different types of the flowers and fruits, vegetables like this. In this slide you are watching the citrus varieties which are different in color, even in the shape also. These are the Japanese varieties, the star ruby which is red in color, and the other one is yellow in color and the red blackish color, and the other part, that is the barley varieties, which is showing differences in their inflorescence. So, even you can watch these sort of the varieties in the nature. In the nature itself they are available, but they are in less frequency. So, now coming to the mutation breeding, you all know that mutations are heritable changes in the phenotype of the organisms. Phenotype of the organism means they exhibit the morphological character changes. So, when there is a change in the genes, then definitely there would be changes in the morphology of the organism. That is why these mutations are considered as a heritable changes in the phenotype of the organisms. So, the term mutation was coined by Hugo de Vries in Enothena lamarckiana. So, these changes are mainly due to the changes in the chemical changes at the gene level. These changes are capable of bringing about new and heritable character variations in crops. If these changes will be selected and used for the establishment of the crop, then it would become a good variety. So, but these mutations occur very low in nature. The frequency of the mutations in nature usually is very rare. That is why they are called as the spontaneous mutations. Actually, the term mutation breeding was coined by Prisleben and Lean, but the first time the report how these mutations would be exploited for the improvement of the crop was given by Muller and Stadler. So, coming to the frequency of the mutations, if we want to increase with the help of the certain chemicals, we can able to increase the frequency of the mutations. So, the rare frequency of the mutation can be increased by the application of the some physical and chemical agents which are called as the mutagens or mutagenic agents, and mutations induced in this way are called induced mutations. So, these agents can be used to induce mutations in croplands, and the desirable variations whatever the farmer want or the customer want, they can be produced in this way. So, the selection of the character is more important in any breeding protocol. So, the approach of plant breeding in which new variations of the crop or new varieties of the crops with desirable characters are developed with the help of the induced mutation is called mutation breeding. So, ultimately the final aim of the plant breeder is to release a new variety with the desirable characters, but here in this protocol the plant breeder is inducing mutations. Mutation, already we told that it is a heritable change. We are making some heritable changes by the induction, by changing the genetic material. That is why it is called as the mutation breeding. So, and in the next slide you are going to know about the process of the mutagenesis and how the mutagens are helpful and their mode of action. So, there are existing only two types of the mutagenic agents, that is physical and chemical mutagens. Coming to the physical mutagens, there are two types of the different types of the radiations available. So, these radiations are classified into two classes mainly based on their energy level. Energy levels are of two types, that is with low frequency and high frequency. So, radiations with low frequency and radiations with high frequency can be employed as a physical mutagens. So, first take the radiations with low energy level. These are capable of causing the excitation at the level of the nitrogen bases of the genetic material, and they are called as the non-ionizing radiation. Their function is only to excite the nitrogen bases, that is the excitation at the level of the nitrogen bases is doing by these radiations. UV light is the good example of the non-ionizing radiations. Then take the radiations with a high energy level. So, when you observe the radiations with high energy level, they caused excitation at the levels of the nitrogen bases only. Their function is only excitation, but when you take the radiations with a high energy level, then they are capable of doing both functions, that is excitation and ionization at the level of the nitrogen bases. That is why they are called as the ionizing radiations. The best example are X-rays, gamma rays, alpha particle, beta particle, and are the good examples of the ionizing radiations. Coming to the chemical mutagens, the chemical mutagens are classified into three categories on the basis of their nature of the action, that is alkylating agents, base analogues and acridine dyes. So, all these three chemical agents perform their function in different ways. Let us watch how these chemical mutagens interact with the nitrogen bases and helps in in the mutagenesis process. Coming to the alkylating agents, they induce the mutation by adding alkyl group to the nitrogen bases. The major alkylating agents are ethyl methane sulfonate, methyl methane sulfonate, ethyl imines. So, they are shortly called as the EMS, MMS and EI. So, their role is just adding alkyl group to the nitrogen bases, causing the changes in the genetic material. So, that is why they are called as the alkylating agent. They are also called as the radiomimetic chemicals. Why they are called as the radiomimetic chemicals means the mode of action of these alkylating agents resembles with the action of the physical agents. That is why they are called as the radiomimetic chemicals. So, they are helpful in the AT-GC transition and GC-AT transitions. Next is base analogues, that is, they are the chemicals which are analogous to the nitrogen bases. So, they can get incorporated into the DNA at the time of the replication and are causing wrong base pairing resulting in mutations. So, they are these analogues are not adding any chemical group, but they are getting incorporated into the DNA at the time of the replication, making some changes. So, the best example is 5-bromouracil and 2-aminopurines. These are the two common base analogues which are commonly used as mutagens. They are doing the ATGC transition, and the other one, the analogues, that is after the base analogues, the most important chemical mutagen is acridine dye. These acridine dyes are the chemicals, they are also capable of getting inserted between the two group bases of the DNA, thus leading to the addition or deletion of single or few base pairs at the time of the replication. If single base pair is deleted or added, or few bases are deleted or added, what would be happen? There is a change in the nucleotide sequence of the genetic material. That is why there is a change in the reading frame of the genetic material. So, the proflavin and acroflavin are the good examples of the acridine dyes. Example is the frame shift mutations. Next, we are moving to the other chemical mutagens also. So, after watching the three categories of the chemical mutagens, there are also certain chemicals which efficiently works on par with other categories of the mutagens. These includes nitrous acid, hydroxylamine and sodium azide. They are also helpful in the AT-GC and GC-AT transition. You know that AT means adenine and it is pairing with the T, and guanine it is pairing with the cytosine, but they are making some replacements. Let us watch in the next slide how these induced mutations are happening. We are watching actually at the molecular level how these mutations could be induced. At the molecular level, induced mutations are doing some changes by the addition or deletion of the nucleotides or nitrogen bases in the genetic material. So, accordingly, they can be classified into three types, that is transition, transversion and frame shift mutations. In the transition mutation, there is a replacement of one purine with another purine or one pyrimidine with another pyrimidine. So, there is a replacement. The purine, you know that what the purines are adenine and guanine, and pyrimidines are thymine and cytosine. If at all you want to replace the adenine, you can also replace this adenine with guanine. So, otherwise if you want to replace the adenine with thymine, so thymine also can be replaced with adenine. If you want to replace the cytosine, then it will be replaced by the guanine. So, the AT and GC can be replaced with their purine and pyrimidines only. Next is transversion mutation. In this, there is a substitution of purine with the pyrimidine. So, one pyrimidine can be replaced with the other corresponding pyrimidines, or sometimes even the other pyrimidines also can be incorporated in the genetic material. So, it is the substitution. So, in the earlier transition, you are watching the replacement of the purine with the pyrimidine with the pyrimidine, but here substitution of purine with the pyrimidine and vice versa, or pyrimidine with the purines. In the frame shift mutation, there is a change in the reading frame. So, if any nucleotide is added, that means A or T or G or C is added at one place, then there will be a change in the reading frame of the genetic material. So, it is causing changes in the production of the amino acids, then the protein material also changed. Likewise, the frame shift mutation causes changes in the reading frame of the genetic material by the addition or deletion of the nitrogen bases. So, next we will move on to the procedure of the mutation breeding after understanding of the physical mutagens and chemical mutagens. So, physical mutagens are radiations and chemical mutagens are the chemicals, that is alkylating agents and base analogues and acridine dyes, even other efficient chemicals, how they would be helpful in inducing the mutations. Now we will proceed to the procedure of the mutation breeding. How a plant breeder do this protocol? It includes the following steps. First is the selection of the plant material and the choice of the mutagen, and the material is treated with the mutagen, and the handling of the mutated population. It would be different in the case of the seed propagated species and clonally propagated species, but the protocol is the same either for the seed propagated species or clonally propagated species. Coming to the selection of the parents, it is the decision on the nature of the variation to be induced and the selection of the appropriate material for mutagen treatment. So, both the nature of the variations to be induced and the selection of the material from the plant must be decided initially. So, based on the method of propagation, seeds and other propagules, that is vegetative propagules like buds, axillary buds or sometimes suckers, can be taken, can be selected for treatment. For in vitro mutagenesis, callus or similar in vitro material is selected. So, this is about the selection of the material. After proper selection of the material, then choice of the mutagen is also plays an important role, because the appropriate mutagen must be selected for the induction of the mutations. Otherwise, the plant breeder would fail if some chemicals would be applied for some material or radiation would be applied for the other material. There are certain principles to follow how to use this mutagen for the selected plant material. Generally, chemicals are preferred for the seed material and radiations are preferred for the vegetative propagules. So, by the use of these chemical and physical mutagens, we are applying the mutations to the plant material. So, the treatment, that is the mutagen treatment, is also very important. There is a principle how much of time is needed to treat the plants with the mutagen. So, initially we want to soak the plant material in water or other chemicals, which is called as a pre-soaking. Pre-soaking is very helpful in the mutagen treatment. After the pre-soaking treatment, later the materials are transferred to the solutions of the mutagen. Here one more point is very important, you wanted to know, that is the concentration close to the LD50 of the mutagen. So, it is considered as an optimum concentration. So, what is this LD50? This is the lethal dose at the 50%. So, in the case of the physical mutagens, actually the source of the mutagen is kept at a safe distance and the treatment is remotely controlled. So, there is a safe mechanism while treating with the physical mutagens. the gamma gardens are there, where the gamma ray treatment is carried out in a protected experimental areas. So, the duration of the treatment is also decided based on the information available. Sometimes it is decreasing, and sometimes the rate of the duration will be increasing for the material. So, what is this LD50 dose? Here is the definition for the LD50 dose. It is the dose of the mutagen that causes the 50% of the mortality of the treated material. So, any mutagen is toxic to the biological system. After this 50% of the concentration, it causes the death and deformities. That is why above the 50% of the concentration of solution, plant breeder must not grow. So, this is the optimum concentration one must follow. That is why this is called as the LD50 dose. Next is handling of the mutated populations is very important. If we are not actually following the that is the characters exhibiting by the mutants, then the final objective of the plant that is mutation breeding will be lost. So, handling of this mutated population and observations and the analysis is also more important. So, after the treatment of mutagens, all this that is observations and analysis part will come. So, next the plant breeder germinate the seeds after the mutagenic treatment, which is called as a M1 population. Generally, mutations are all recessive and most of them are selected in later generations, but the dominant and pseudo dominant alleles are selected initially in the M1 itself. These M1 plants are selfed and the seeds are collected separately, and they are used for the next progeny, that is called as the M2 generation. So, the M2 generation is grown next for their progeny, which is from the M1 seeds. Oligogenic mutations are selected at the in this population only. So, the M2 population is more important to observe any variations are found in the crop plants. So, if any variations are found in this M2 plants, then they are treated as the mutant plants, and these are isolated after necessary trials. So, from this chart you are going to know about how the breeder is going for the trials. This is the first year where the seeds are spacely planted. In the second year, individual plant progenies grown from the M1 plants. In the M3 generation, individual plant progenies are grown, but the superior mutant lines are harvested in bulk if they are homogeneous. Otherwise, we will be moving to M4 generation, but in many cases in the M3 generation itself they will be bulked. So, after the M3 generation, the preliminary yield trials are conducted in the M4 itself. In the M4 generation, if the yields are fruitful, then we are moving to the M5 generations, that is coordinated yield trials are carried out till M6 and M7 generations. If the characters are consistent and they are desirable characters are founding continuously in all generations, by M8 or M9 the lines will be selected and released if they are promising in growth and yield. This is the main objective of the mutation breeding. If they are not sustaining well, again the experiment have to be repeated again. So, that is why this mutation breeding is somewhat highly risk and laborious. So, in case of the polygenic traits, the inferior plants are rejected at M3 and M4 levels itself. Later the remaining seeds are bulked as usual to the M5 and M6 and M7 generations for yield trials and coordinated trials. So, by M8 and M9 generations, it will be released as a new variety. This is in the case of the seed treating crops. Coming to the vegetatively propagated crops, it is somewhat different. So, here is one more slide for you, where the preliminary yield trials for suitable checking, how it is going on in M4 in a separate lines. So, superior lines are selected. Each blue color rectangle is representing each line selected from the previous progeny. From M5 to M7, replicated yield trials, that means they are divided into replicas, and outstanding lines are released as a new variety. So, in M8, seed multiplication are done. So, in the vegetatively propagated species, the procedure is same, but the mutations are expressed as chimeras. So, these are take the vegetatively propagated crops like chrysanthemum or sometimes dahlias. They are existing in different beautiful colors. These are called as the chimeras. So, the chrysanthemum exhibits in various colors, like yellow color is there, pink color is there, white color is there, sometimes dotted colors are there. So, these are all different in colors. Even the fruits are also different, but fruits comes from the seeds, that is why that case is different. But take the vegetative crops like chrysanthemum, jasminium, like different types of the flowering plants like tulips or roses. You can take the examples for the vegetatively propagated plants. The same thing, you treat the vegetatively propagules with the mutagen, that is called as the VM1. In VM2, you observe the chimeras whether they are sustainable or not. In M5, conduct the yield trials. From M4 onwards, preliminary yield trials. M5 to M6, M7, conduct the coordinated field trials, and by the end of the M8, that is VM8 or VM9 generations, it will be released as a new variety. So, today in market, several varieties of the flowers are available, and they are also changing colors from years to year. The original existing material will be with the plant breeder itself. Next is applications of the mutation breeding. So, now you watched about the that is the mutagens. So, mutagens, how they are able to induce the mutations, and the process of the mutation breeding, how the plant breeder do the protocol from M1 to M9 generations by conducting preliminary yield trials and coordinated yield trials. So, mutation breeding can be used to develop the improved cap varieties. There are several resistant varieties like disease resistant variety and frost resistant varieties and temperature tolerant varieties and stress tolerant varieties. Likewise, several crop varieties have developed by the plant breeders in rice, maize, jowar. Take any crop which is economically important, not only in the cereals, even in the pulses or any important commercial crop, according to the objectives of the and the necessities of the market. The plant breeder takes the and fix the objectives and improves the quality of the crop. Next is to induce the male sterility. The male sterility is one more important factor in the plant breeding, which helps in the creation of the male sterile lines. So, it suppresses the growth of the other that is the part of the gynoecium, and allows the androecium to give only pollen grains, called as the male sterile lines. So, production of the male sterile lines not possible in the nature, but they are very rare, but through the mutation breeding it is possible to produce the male sterile lines for the production of the haploids. Haploids are usually produced from the pollen grains, because the pollen grain is the haploid cell. If any regenerants would be produced from the pollen grain, they are called as the haploid plants. In nature, haploid plants are actually it is very difficult to produce, but through mutation breeding it helps in the production of the haploids also to create additional genetic variability. So, the main target is additional requirements for the crop. So, it would be produced by the variations in the crops. So, this mutation breeding is one of the step which would be able to produce the genetic variability. And the other objective of the mutation breeding is also adaptability of the crops to various environments. If we introduce the crop in one environment, if it is not sustained with that environment, the main theme of the breeding would be lost. So, all the 9 to 10 years of the efforts would be will be wasted. That is why the adaptability of the crops is also more important in the mutation breeding. So, this is important, some of the important objectives, that is the characters like higher yield, short structure, earliness, stress resistant, and bold, that is seed size is also more important. These are the crops of the mutant varieties developed in barley, pea, groundnut and rice. So, you can take these as examples. The merits, coming to the merits of the mutations, mostly they are all undesirable and there is no of value to the breeder. Many mutations are lethal, sometimes the 50 dose is not suitable, then the plants would be dying. So, that is why all the mutations sometimes may be lethal to the plant material. The mutations rate is also extremely low, then we have to improve the dose. Then we are discussing that the dose will be somewhat that is fatal or lethal to the organism. So, that this is also one of the limitation in the mutation breeding, and it is highly difficult to tolerate with the dosage of the mutagen. The stability of the mutant is also thoroughly tested by the preliminary yield trials and coordinated yield trials. So, otherwise these mutants have a tendency to revert. This is the serious lacuna in the mutants, that is their characters would be reverted if they are not properly checked out. That is why the preliminary trials and coordinated yield trials are highly essential for the plant breeder to check out the qualities, whether they are remaining in the generations together. Next is mostly induced mutations are recessive, that is why they have to be induced in a double dose to be expressed phenotypically. These are certain demerits of the mutation breeding. From leaving out the demerits of the mutation breeding, the mutation breeding protocol is one of the highly sustainable and efficient protocol for the plant breeder when there is a loss of hybrid vigor, or sometimes the crop variations are not fetching out. Then this is only the final step by the plant breeder to improve the crop varieties. Today, I would like to make you know about the use of RFLP and RAPD in plant breeding and crop improvement. So, these are the two markers in the plant breeding technology. So, recent advances made possible to identify the accurate genetic polymorphism. All these technologies enhanced the discovery of the genetic markers. The genetic markers are heritable differences in the nucleotide sequences of the DNA. Nowadays, there are available different types of the markers like morphological markers and molecular markers. We are discussing only molecular marker, that is DNA marker in this class. So, there are two types of the DNA molecular markers are available. They are non-PCR based approaches and PCR based approaches are there. In non-PCR based approaches, RFLP is there, PCR based approaches, several molecular markers are there, RAPD, AFLP, SSR, SCAR, STMA and SNP etc. are available today. Now coming to the RFLP, that is the RFLP is the restricted fragment length polymorphism, which is developed in 1980s by the Alec Jeffrey. So, it is based on Southern blotting technique. It is used to analyze the variations in the length of the fragments that is developed from the DNA shearing. So, to watch the variations in the DNA, the restriction endonucleases are used. The same endonuclease will be used for the different organisms, but the restriction endonuclease makes the cuts at restriction sites. So, the restriction endonucleases and restriction sites plays an important principle in the RFLP, that is restricted fragment length polymorphism. It detects the differential movement of the bond on the gel lanes. So, it is the first molecular technique developed based on the hybridization. So, plants are able to synthesize their DNA or amplify their DNA with high accuracy and rapidly, but at the same time mechanisms are also available which can make the changes and they are operative at the same time. Simple base sequences or sometimes large scale changes like deletions, inversions and translocations or transpositions may occur in the genetic material. All these lead to the gain or loss of the restriction sites, which is leading to the restricted fragments of the genetic material, that is restricted fragments of the different length. These restricted fragments of the different length can be detected on the Southern blots using a suitable probe. So, they are actually detected by the differential movement of the bond, that is band on the agarose, that is on the gel lanes collected from the different strains or species. The main steps involved in the RFLP technique is isolation of the DNA. After isolation, we have to cut the DNA using the endonucleases. Later, we separate those DNA fragments by using the gel electrophoresis. Then they are transferred to the nylon or nitrocellulose paper, which is the material generally used in the Southern blotting technique. Afterwards, visualization of this special DNA can be done by the radioactively probed, that is radioactively labeled probes. So, finally, the analysis of the results are done. So, based on the principle, that is the restriction endonuclease principle, that is which makes the cuts at the restriction sites and makes the different length of the fragments, because these restriction sites for a particular enzymes are present throughout the genome at several points. That is why the different length of the fragments are obtaining in the DNA digestion. This is the principle of the main principle of the RFLP while doing the technique. Next is that is this is here RFLP technique, where you are observing the different types of the bonds. That is in the normal case you are watching the two bands, whereas in the disease case it is a special band which is displaying in the Southern blot diagram. So, after gel electrophoresis, when it is radioactively labeled probe that is detecting the gel that is the RFLP band, then the one more band is there in the diseased causing strain. The RFLP technique is here is also one more diagram for you where the whole technique is running. And coming to the applications of the RFLP, we are able to isolate and identify the any type of the gene, and it will be linked to the RFLP locus. What is RFLP locus means? Each band, that is RFLP band developed in the RFLP technique, is called as the RFLP locus. So, these genes would be linked to the RFLP locus. It is a very simple technique and it does not require any sequence specific information. It is a codominant marker, that is why it is used in the differentiation of the heterozygous and homozygous individual. So, it is a supplement to the regular plant breeder as a indirect selection, and it is also used in the detection of the diseases. Coming to the limitations, large amounts of the highly pure DNA is required for the RFLP technique. It needs the good supply of the probes. So, with probes only reliably detecting the variation, that is why we have to give good supply of the probes. Without the good supply of the probes, there will not be detection of variations. That is why there is a need for the good supply of the probes. It is a laborious and expensive process because it is difficult to identify a suitable marker for the genomic DNA or cDNAs. It is a time consuming because it is not amenable to automation. It requires lot of expertism because it is there is a autoradiography procedure where there is a use of radioactively labeled process. So, that is why the RFLP, there are applications that is advantages are also there and limitations are also there. However, it is the one of the efficient protocol to detect the polymorphism. Mainly it is used for the analysis of the genetic diversity of the populations or related species. That is why the RFLP is variously used by the several plant breeders. The next technique is RAPD, that is restricted amplified polymorphic DNA. This DNA marker is also useful in the plant breeding. So, these markers, this technique is actually utilizes the single short oligonucleotide primers of the 10 nucleotide base sequences for the RAPD technique for the amplification of the set of DNAs which are randomly distributed throughout the genome. That is why the name itself RAPD means randomly amplified polymorphic DNA. So, these primers are used to amplify the DNA which are present randomly throughout the genome. So, in the amplification process actually generated from a region which is flanked by a 0 base primes, that is 0 base prime region, that is the priming sites, and all the different genomic materials would produce the different amplification products, but the PCR amplified product is generated only one for one individual, not for others. So, this is the technique, that is RAPD polymorphism among the land races of the sorghum, that is OP, that is these are the some of the RAPD primers having the 10 base sequence, and this is the that is gel configuration on the agarose gels. So, this is a RAPD banding pattern comparing with the some ladders taking from the lambda DNA like that, and likewise RFLP, the RAPD also possesses the same steps, that is like isolation of the DNA, selection of the, that is cutting of the DNA, selection of the DNA on the agarose gel, but there is a transfer of the fragments on the ethidium bromide. So, ethidium bromide is a dye by which that is the RAPD's amplified products are visualized under UV light. So, there in RFLP we are transferring to nitrocellulose paper, here we are visualizing under UV light. So, the RAPD applications are These are construction of the genetic maps and helpful in the mapping of traits and analysis of the genetic structure of the population, fingerprinting of the individuals and identification of the somatic hybrids. Finally, it is very useful system for the characterization of genetic resources. There are certain advantages when compared to the RFLP. It is a simple, quick to perform. The RFLP technique requires high amount of DNA, whereas this technique requires only small amount of DNA. It involves no radioactivity, that is why no expertism is also not required. Coming to the limitations, it is unable to distinguish between homozygous and heterozygous. Actually, coming to the inheritance, the RAPD band actually inheriting from parents to offsprings as a dominant marker. That is why if the band is present, it corresponds to the RAPD dominant allele. If the band is absent, it corresponds to the recessive allele. That is why the RAPD is unable to distinguish between heterozygous and homozygous organisms. The RFLP is able to distinguish between RAPD that is homozygous and heterozygous individuals. This is the comparison about the important characteristics. You can go through this slide, and these are the references which you would like to know. You want to further read about the RAPD and RFLP. My next topic, I would like to give information about the role of the somaclonal variations for crop improvement. The presence of genetic variability that is present in the cultured cells or plants derived from those cultured cells or progeny of such plants is called as the somaclonal variations. In tissue culture, the plants derived from the cell culture or tissues are called as the somaclones. If variations are present in the somaclones, they are called as the somaclonal variations. So, simply the variations present in the somaclones are called as the somaclonal variations. The term was first used by the that is coined by the Larkin and Snowcroft in 1981, but the first report of morphological variations from the cultured cells, types, that is based upon their features, that is based upon the derivation of the tissue. There are gametoclonal variations, variations observed from the plants that is derived from the gametic cultures. They are further divided into androclonal and gynoclonal variations. If variations are found from the plants derived from the pollen cultures, they are called as the androclonal. If they are derived from the ovary culture, they are called as the gynoclonal variations. Calyclones means the variations are found from the progeny of the plants derived from the callus, likewise mericlones, protoclones. If they are from the protoplast culture, they are called as the protoclones. However, all these somaclonal variations are heritable mutations and they persistent in plant population even after plantation into the field. Coming to the mechanism of the somaclonal variations, all these variations are either transient or genetic. Transient means they are epigenetic. If they are genetic means all these variations are carried to the next generations. That is why these variations are more important in the crop improvement. In the genetic variations, already the pre-existing variations are present in the somatic cells or the explant of the tissue culture. Epigenetic means they are present in the earlier culture of the system. system, but usually they last when the regenerants are reproducing sexually. Origin of the somaclonal variations, how these variations are observed in the plant tissue culture. So, these variations are actually either oligogenic, that means they observed for the single trait or more than single trait at a time. They may be occurring either one of the following regions. That may be the first region is chromosomal abnormalities. You know that the chromosomal abnormalities occur due to the changes in the chromosome number or the chromosome structure. These are some examples, that is euploidy or aneuploidy, deletions, inversions, duplications, translocation. All these chromosomal abnormalities causes the genetic changes and makes some changes. The gene mutations, that the gene mutations are also one of the reason for the change in the genetic material. The plasma gene, that is the change in the plasma genes, activation of the transposable elements, DNA methylation, and the rearrangement of the cytoplasmic genes alters the genetic material in the culture conditions. Coming to the advantages of the somaclonal variations. So, the origin of the somaclonal variation may be either spontaneously or sometimes it can be induced, like mutation breeding. But the advantages when compared to the mutation breeding, there are certainly the somaclonal variations shows advantages far more better than other breeding methods. The frequency of the variation is high under in vitro conditions when compared to the spontaneous mutations or induced mutations. The time is also required very less in release of a new variety when compared to the other protocols like hybridization and selection. Even it reduces the that is the unique mutations. In tissue culture, unique mutations occur and which could not be possible, that is in the tissue culture, which is possible, it may not be possible during the crossing over as the germ plasm is not available. The pedicel, that is jointless pedicel tomata mutant is in tissue culture only produced. It is the good example for the somaclonal variant. It occurs for a trait either from the nuclear or that is a cytoplasmic material. Creation of the additional genetic material is also possible. There will not be any drastic changes in the genetic material. Next is it saves time by reducing the prolonged procedures of the hybridization and selection. It is a highly efficient protocol because it screens a larger number of the clones at a time within the short period of time and with less time and labor. So, it is simple and the cheapest form of the plant biotechnology when compared to the plant that is somatic hybridization and transformation. Variations can be selected at cellular level only in somaclonal variation, which is not possible at the plant level. Coming to the disadvantages of the somaclonal variation, a serious disadvantage occurs in the operation, that is the exploitation of the tissue, that is in vitro propagation or micro propagation methods, where the main objective is to produce uniform true to type individuals. So, there is a limitation with the somaclonal variation where the changes continuously happening in the cell culture system, and sometimes it leads to the undesirable results like poor performance, poor growth is there, sometimes the fertility rates are also reduced, and it requires extensive field trials, and extended field trials are also necessary for the somaclonal variations, whether the variations are continued for generations to generations, and no suitable even the complex agronomic traits like yield, quality may be checked after the long procedures of the tissue culture. So, these are all the limitations, and even the variations which are produced in the somaclonal variations may not be useful for the plant breeder, and sometimes they may be lethal because these are all pleiotropic effects, because they are not considered as a true variants. That is why the somaclonal variations are sometimes may not be useful for the plant breeder. There is that is reduced or no regeneration capacity of the resistant clones. So, the somaclonal clones, the somaclones mainly they are of reduced growth generation by generation, and even finally the regeneration capacity also lost in the cultures. That is why they are considered as the poor regenerants. Applications are applicable to only those species whose cell cultures regenerates complete plantlets, then only the somaclonal variations would be useful. Otherwise, if they would not be regenerated into new plants, the whole that is aim of the somaclonal variations will be useless, and they show some undesirable genetic changes like reduced fertility, growth and poor performance. These are the main limitations of the somaclonal variations, and the phenotypes expressed in the selected lines may not be expressed by the plants regenerated by them, and the small and viable portion of the clones are only stable, and the larger portions of the clones are usually unstable, and they are not further used in the continuous cultures. That is why they will be discarded in the subsequent cultures. Only few cultures are taken for the next regenerants, and most of the variants are not at all novel, and they are not useful for the plant breeder. There is a poor correlation between the that is glass house and field performance of the somaclonal variants. So, that is why also the analysis of the somaclonal variants, they do not give good results, and the superior varieties like agronomical traits by screening of the progeny of the tissue culture or regenerated plants only can be done. Finally, coming to the application, there is a increased and improved production of the secondary metabolites. Not the complete plantlets of the regenerants is not possible through the somaclonal variation, but there is a increased and improved production of the secondary metabolites. Selection of the plants resistant to the various toxins, herbicides and salt tolerant and mineral toxicity important resistant tolerant plants can be produced through the somaclonal variations. That is why it is suitable for the breeding of the plant. Here is a slide showing the examples of the somaclonal variations in some crops.